today is Dr. Carrie Waterman. She's a research scientist at Rutgers University in the Department of Plant Biology and Pharmacognosy. And she's working in the areas of agriculture and nutrition, and she has a lot of experience working in Africa. And today she's going to talk to us about moringa. So with that, I'll turn it over to you.
Thank you so much. Thank you all for having me today. So I'm just going to talk a little bit about the research that I've done with moringa, and then my KO1 grant, which will have me doing research in Kenya over the next 5 years with moringa. So I'll just go through how I kind of got interested in moringa and then talk about its nutrition, medicinal properties, some of the phytochemistry that's in the leaves that's responsible for what we believe the medicinal properties to be, how we've worked on different extraction processes for the moringa leaves and its biological activity, and then a little bit about the KO1 grant, and then some additional opportunities with moringa that I see, particularly at UC Davis, between this intersection between agriculture and nutrition. So I was a Peace Corps volunteer in West Africa in 2002, and this is where I was introduced to moringa. My primary objective was a math and science teacher, but I spent a lot of time with the traditional healers and visiting some of these sites where they treat all of their patients with plants. And so moringa was really advocated by the Peace Corps as this multi-purpose plant and promoted by the WHO as well for its health uses. So we did a lot of training, particularly focusing on nursing mothers and infants, and the WHO has sponsored some research to fight malnutrition in infants. And then there was also a lot of agricultural projects as well. So moringa is in the order Brassicales, which is where you find other cruciferous vegetables like broccoli and cabbage. And it grows, it's native to India, but it grows really in any tropical, subtropical area. And as long as it doesn't freeze in that area, you can grow moringa. It also thrives in really poor, nutrient-depleted soils and grows really fast. So it's a really good plant for a lot of areas where they're facing drought and other things. So it's a multi-purpose plant, and a lot of its parts can be used for different things. The leaves really was what I've been focusing on for its nutrition and medicinal properties, but other really interesting parts are found in the seeds. They can be used for water purification, and I'll talk about that a little bit. And the roots as well, and the bark. And so the other part that's eaten a lot is the pods, the immature pods. And so in Indian cooking, they're often used in sauces as well. They're kind of like green beans. So moringa meets a lot of basic nutrition requirements in terms of macronutrients and micronutrients, but then it also has a plethora of secondary metabolites. So that's really been my focus, and my expertise is in the secondary metabolites that provide additional benefit. And these are just some dishes and the way the pods have been prepared or the leaves have been prepared. You can also make a tea out of it. So it's been promoted by the WHO and a lot of different organizations because of this high nutrient content. And this is partly because moringa leaves don't contain as much water. So most vegetables are about 90% water, and moringa is only about 80%. So you get these much higher values of a lot of these nutrients. And then this is just a breakdown if you guys are interested in any particular nutrient. Kay had asked me to put it in terms of, this one is just per 100 grams. And comparing it to spinach, this would be fresh leaves. And so you can see high mineral values. It also has all of the essential amino acids as well as flavonoids. And then this is comparing it per 100 calories, but you can see that the amount is much less that you would need to eat to get that same amount. And then this was an article put out looking at using moringa leaves to meet the nutrient needs of 1 to 3-year-olds. And so this is basically 5 grams of moringa leaf powder. So this is usually how it's made. It's fresh and then they'll dry it. Or a cup of fresh moringa leaves. And this is basically the percent they're recommending that you would, the percent of the nutrients needed that you would get out of that. So it's really, really good for iron. You know, you can see you can get about 23% of your iron just from 1 tablespoon of moringa leaf powder. So you can basically mix this into the foods or the sauces after it's been cooked. So that's kind of the nutrition part that I haven't really done as much work on, but I think that you know, in the future, combining, I think that there's, you know, to prevent a lot of diseases, having good nutrition is essential. And especially in Africa, if you're well-nourished, you're much less likely to develop things like malaria as well as metabolic disorders like diabetes. So moringa has been used for the treatment of inflammation, diabetes, and cardiovascular disease, and also just for the promotion of well-being. So in 2012, I took a postdoc position at Rutgers, and they were focusing on botanicals to treat metabolic syndrome. So this metabolic syndrome is this kind of combination of risk factors, including diabetes and obesity. So you have things like high blood glucose, high blood pressure, high triglycerides. And then the other thing that they're really noticing with metabolic syndrome is that there's this low-grade chronic inflammation, and that's associated with almost every disease state. So acute inflammation is good when your body needs it, but when you have obesity and diabetes, you're constantly making these inflammatory cytokines. They were also working a little bit about the role of gut microbiota in obesity and diabetes. So these were some papers that came out from Jeff Gordon's lab about basically the alteration of certain phylyl groups once you have obese mice and the genes in there. So we were trying to see, this was another paper that showed that basically what they did was they had germ-free mice, and they transferred the gut microbes from obese mice to these germ-free mice, and they showed that those germ-free mice that got the bacteria had a much higher increase in body fat mass, and that they were able to harvest more energy from the food that they were eating. So meaning if you're obese, you actually extract out more calories from the food you eat, which just perpetuates the problem. So we were a little bit interested in looking at microbes, and this is kind of how they see this effect happening. As you have high-fat feeding, it changes the microbiota. You get increased permeability in the gut, and so you have LPS and other markers that cause inflammation, and then this low-grade inflammation can lead to metabolic disorders. So what we're hoping is that by administering polyphenol-rich foods, which moringa is one of these, both have antibacterial and anti-inflammatory properties, that you can then, you know, decrease these alterations in the gut microbiota, decrease inflammation, and hopefully decrease weight and insulin resistance. So this was a really new field to me. I had been working before with isolating compounds and testing them for things like anti-malaria or anti-helminthic properties, and now we're in this environment of foods that are very complex in the nutrients that they offer, the intestinal environment, the process of inflammation, and then the microbiome. So I introduced moringa as a potential polyphenol-rich food that we could work with at Rutgers. So these compounds are found in many plants. Camphorol glycosides, chlorogenic acid, and quercetin molecules, and these have been shown to have a really positive effect on metabolic syndrome. But as we began to look at moringa a little more closely, I found that there were actually even more unique compounds in moringa, and these are the ones that are related more to cruciferous vegetables. So they are in a precursor form called glucosinolates, and then what happens is there's an enzyme myrosinase that's stored separately in the vacuoles of the plant cells. And then if an insect chews them, you basically cleave off this sugar, and you're left with this thiocyanate group, which is very reactive and very spicy. So it's like what they call the plant releasing its mustard bomb in defense of that insect. So it tastes pungent, and that's what you taste when you eat arugula. That's what you smell when you overcook broccoli. You're smelling that sulfur. So most isothiocyanates, which is this form after the myrosinase is cleaved, are really unstable. But in the case of Moringa, and it has 4 forms of these where you have monocetylated groups here, you have this additional rhamnose sugar after the cleavage, and this makes it very stable. And this is almost unheard of in the natural world. So this is just comparing broccoli's glucoraphanin, and then basically if you eat fresh broccoli, you can get some conversion to the isothiocyanate, which is the bioactive form. But not everyone has those enzymes in the bacteria that live in their gut, and it's very unstable and rapidly degrades. So it's hard to make a dietary supplement that contains high levels of sulforaphane. But in the case of Moringa, as I said, you cleave off this sugar and you're left with another one that's very stable. So we did some stability studies, and you can even see that when you isolate these compounds, they're actually a white powder versus sulforaphane, which is an oil. So these are different these are 2 of the different isothiocyanates, MIC1 and MIC4. At 25 degrees, they remain fairly stable. And at 37 degrees, you get some degradation from MIC1. But compared to what's reported for sulforaphane, you basically lose everything within 5 days. So basically, MICs could provide a source of a strong bioactive compound that you could put into a nutritional supplement. So the classic way that Moringa is grown and produced as a powder is they basically dry and then crush it. So they'll lay out the leaves, let them dry, and then pound them into a powder that you could add to the food. But what happens is when you dry them, you destroy that myrosinase enzyme that converts the glucosinolate to the isothiocyanate. So what we proposed is actually take the fresh leaves, crush them, pound them up, get that glucosinolate to convert, and then you'll have high amounts of isothiocyanates. So we did a couple different experiments. We looked at some commercial products of the isothiocyanate concentration, and they're all pretty low. We also looked at not adding any water, which would be amenable to processes that we can do in Africa, and we get much higher values. And then, of course, if you extract with water, you can really get higher concentrations of these. So I'll just talk a little bit about the activity of MC, which I say is the Moringa concentrate, which is basically this extracted and filtered leaf powder. So we did some studies on looking at inflammation, because as I said, this is kind of an underlying cause in a lot of different diseases. And so when you take the Moringa concentrate, we looked at 2 cytokine markers, INOS and IL-1 beta. And so you can see it has a nice dose-dependent reduction in the expression of this RNA. And at 100 micrograms per mil, this basically contains between 1 and 5 micrograms of MICs. And if you purify those compounds and test them against the same markers, you also see significant reduction in these cytokines, suggesting that they're really contributing to this anti-inflammatory activity. And these are also micromolar concentrations is what you want. A lot of things are active, like polyphenols and stuff, but you're really not quite at those low levels. This is more in a range of really what you would see with a drug. From there, we then proceeded to do an in vivo study with C57 mice. So this is where you induce them to be obese by feeding them a very high-fat diet, so 60% fat in the diet. And then they also develop type 2 diabetes or something similar to it. And so we gave them, basically combined this Moringa concentrate into the food and then fed it to them for 12 weeks. And what we saw was really impressive was that this is the control group, the very high-fat fed diet, and then the group that had the Moringa concentrate in it. So they still gained weight, which is what a mouse would do, but what's really reported if you were just on a low-fat diet would be here. So we were able to reduce that weight gain by about half. And if you look at the fat mass accumulation that they had, and this is why moringa is really good for enhancing the lean mass or the protein level. So this is at 4, 8, and 12 weeks, the fat mass as a percent of body weight was much lower in the moringa-fed group. And then if you look at free fat mass, which is basically lean mass, that continued to increase. So that also shows that they weren't, that there weren't really, I mean, we have to do follow-up studies to make sure there's no toxicity, but basically they were still gaining weight and still gaining muscle mass, and there were no other indications of any toxicity. Looking at oral blood glucose levels, this was done at 2 weeks. So this was just comparing this to metformin, so it was able to reduce the blood glucose levels. And then we also looked at the end of the study at a lot of biochemical markers, and so in terms of diabetes, lower insulin, leptin, resistin, inflammatory markers, although the TNF was not significant, and then cholesterol and slightly lower triglyceride levels. So the problem is that because they didn't gain as much weight, you don't really know what's cause and effect here, right? So I could show this same data for lean and obese mice. So I don't know the mechanism of how moringa actually acted in these animals, but there were definitely positive effects, and this provided really good preliminary data for more grant applications. This also looking at inflammatory markers in the liver and the ileum and the adipose tissue, we saw lower TNF values as well. This is a little graphic, but representing their livers. So this was the very high fat fed diet where you can see basically fatty liver disease and deposits of the fat, and you can see that here as well. And then this was a representative liver of the 5% moringa-fed mice. So, what is moringa doing in these mice? And there's all these variables, and that's the problem when you work with animals and not just cells. And I'm sure, as you guys all know, when you work with people, it's even more complex. But what we thought was maybe it's having some effect on glucose metabolism. So this was an in vitro study that was done in liver cells, and what we found was that moringa inhibited gluconeogenesis quite significantly, and then these are the pure compounds, the MIC1, MIC4, and then this is sulforaphane from broccoli. And broccoli has been researched quite a bit for cancer prevention, but there really hasn't been any work on broccoli with diabetes, and I think that that's also an interesting avenue. As much as I'm promoting moringa, it's much easier probably to get Americans to eat broccoli. It's something that they know versus moringa. But these compounds are really active. But again, with sulforaphane, you face that problem of the only way to really get it is through fresh broccoli. So this is one kind of hypothesis that we want to look at more. And then the other one was that is moringa having some thermogenic or lipolytic effects? Basically, is it promoting the breakdown of lipids in the body? And so we looked at the adipocytes from these mice, and you see upregulation of some of these lipolytic genes. However, UCP1 is usually, we expected that to be much higher, but there is some interplay between some of these. So UPC1, like with capsaicin and peppers, that's what you see a real increase in that it's in the brown fat tissue, it's burning a lot more fat. And then we also did another really short-term 2-week study where we put the mice in metabolic chambers, and we showed that there was a decrease in the respiratory exchange ratio, pretty slight if you actually look at the scale, although it was significant. And this also indicates that there's more burning of fat than carbohydrates. So as you do more experience, you just have more and more questions, really. And so we were able to apply for a lot of different grants and funding on moringa. So out of Rutgers and then Pennington Biomedical Research Institute in Louisiana, they do a lot of clinical studies on obesity and diabetes. So we have a couple different things going on in terms of kind of business. We do have a patent on the moringa extraction, that very simple thing of just crushing it first and converting and getting high concentrations of isothiocyanates. So we're looking at this for both nutritional supplement development as well as even cosmetics. So the hope is to try and make some profit that can then also come back to the research avenue of things. So we have a small, an SBIR grant from NIH to look at the treatment of intestinal bowel disorders and then also further the development of the product. And we have some interest from L'Oreal right now for a skin cream because inflammation, again, in the skin is also relevant. And then we also have the NIH has a botanical research center grant between Rutgers and Pennington, and so they're also looking at metabolic syndrome. They're going to look at things like the microbiome, bioavailability of these compounds, how do they actually break down in the body. And then what I was awarded is this NIH Fogarty K01. So that's for international career development, and then I'll be focusing on the production and processing it without evaporation techniques because I'll be, in the villages growing these things, and then also looking at metabolic syndrome. So basically, I'll be implementing the botanical production and processing of it, and so I leave tomorrow for Kenya, and so I'll be meeting with people that Beth Mitchum from the Horticultural Innovation Center, she spoke last week. She funds a project there where they're already working with some villages that are growing the moringa, so if I can use them as a source, that'll be great. And then we'll also do some of the analysis at Kenyatta University of, does the moringa that's grown in Kenya have these same levels of isothiocyanates? And then do some in vitro work at the Kenya Medical Research Institute, and then really work on coordinating this kind of interdisciplinary team, and that's part of what the K is about as well for career development for myself. And then right now the grant is through Rutgers in the States, but what I'm hoping is that by January I can actually transfer the Rutgers portion to UC Davis in this department. And I think that will be great because there's a lot of experience of people working in Africa, and eventually, because moringa has been eaten for so long, I'd much rather be feeding it to humans than to rats, and I think that's just much more relevant. So that's kind of the plan, and after I left Rutgers, I took a job at Bayer Crop Science in West Sacramento. They used to be located in Davis, and so they're kind of on my way too, and they're interested in working with smallholder farmers in Africa as well. So there's a lot of potential, and hopefully good things will come. So I have 5 years to work on this grant. And then more specifically, the three aims in terms of the scientific aspect of it is to develop a cost-effective moringa supplement concentrated with both nutrients and then also these isothiocyanates, which I believe to be the primary contributors to the anti-inflammatory and anti-diabetic effects. And then look at these in pathways of cellular protection, inflammation, and then also metabolism. And then the third is to evaluate MC's ability to really, this is going to focus in on type 2 diabetes. So I'm hoping to actually use a rat model here at UC Davis to look at the type 2 diabetes development. And then so that's kind of my grant, and that's what I'll be doing for the next 5 years. But while I'm in Kenya, I think there's also a lot of opportunities, as I was saying, to kind of look at this agriculture and nutrition combined together. So there's already some projects going on in this department in Kenya, and as I said, with the Horticulture Innovation Center on the promotion of green leafy vegetables. I've also talked with Howard Shapiro, who has a program where they're looking at the 100 most consumed vegetables in Africa and doing genetic sequencing on them. So if those samples are there and I can meet with their colleagues and see what the isothiocyanate concentration is, we can also maybe hone in on some of the best varieties that have these compounds. And so I'll just talk a little bit more about moringa and some other ideas that we might be able to pursue in Kenya that kind of broaden out beyond just my kind of phytochemical analysis and testing in these things, is that it can also be used as an animal fodder and growth promoter for cows and chickens, and also in increasing milk production in these animals. And then I had alluded to this use of the moringa seeds for water purification. So this is a pretty neat technique. They basically dehusk them and then put them down to a powder. And there's proteins in the moringa seed extract that act as flocculating agents. And so they're basically charged molecules that will bind the soil in the water and then sediment down. And so any soil particles in the water, that's usually where you would find bacteria. So they say that you can get down to like 99.9% potable water from these seeds. So this is, pretty amazing and very important in the developing world. It hasn't been my focus, but my dream is 20 years to have a Moringa Institute, and this would certainly be a major part of that. It can also be used as biodiesel. So that's another effort. The seeds are very rich in these oils, and that's also used a lot in cosmetic products. And it can be used as a biofertilizer, nematocide, and growth promoter. So people basically are doing this, where they're planting moringa in rows in between their crops. And then because they're nutrient rich and these compounds in the seeds and the roots are also preventing nematode growth. So there's been some publications on that. And so basically overall my goal is to work both with universities in Kenya and in the US to work with industries such as Bayer and then also just research institutes to kind of foster collaboration and really ultimately improve the lives of Africans in some of these villages, both in terms of income and then also nutrition and health. And so I'd just like to thank some of my collaborators at Rutgers, at the Pennington Biomedical Research Center, and then the grants that have helped fund the research. And with that, I'll thank you. And if you have any questions.
I have two questions. One is, do you know about anti-nutrients with moringa? Is it high in phytate or any other anti-nutrients?
With the oxalate, is that what it is that you asked me about? There isn't. It doesn't have any of that, but I do believe it does. It has tannins, so there's going to be some anti-nutrient properties.
Next question was, when in the study where there was moringa was given to rats with a high-fat diet, was there a different amount of fat? Like, did it displace any of the food that was given to the two groups?
I'm not following. So,
were there two groups, one that got moringa, one that didn't?
Yeah.
Did both rats get, or both groups get the same amount of diet?
Yeah, so they do
just
it
added extra moringa?
Per calories, yeah. So they calculate how much fat and protein you would get from that moringa, and then they balance it out per calorie.
So it displaced some of the food of the other fat.
Yeah, but per calorie, I think if I have it, what you're getting is the same. Let's see if I can find it. If I'm following your question. Let's see. So in terms of grams, this is how they did it between the two. This was the very high fat diet, and then this was the 5%. So you end up with the kilocalories you get is the same, and the fat that you get is the same. And the carbohydrates.
What was the diet of the group? What was the food?
It's 60% fat, I think, from lard, and then I'm not sure the other components.
So it's super nutrient poor.
It meets all the basic nutrients. They add in nutrients.
Oh, they add in
well.
nutrients. Okay.
Yeah. But then this was only balanced out for protein, carbohydrates, and fat.
Carrie, I'm curious have you ever found in your research that too much of moringa can it have a toxic effect or negative effects? Because there's so many positive effects here, but just wondering if there was like a saturation or tipping point.
Yeah, and they have done studies. I mean, I think any compound that's effective should be toxic at some level because otherwise it's just, so there is kind of, a sweet spot with that. And I think that the leaves, because they've been consumed for so much longer, are probably safe. It would be hard to, I think, kind of overdose or take too much of the leaves. But the seeds are now being promoted as a medicine too, and they have compounds in them. They have some alkaloids and stuff that I think could be toxic. But even the leaves, they've done some toxicity studies, and it's, you know, what you would take as a supplement or a food is way beyond what you would recommend. But yeah, that's definitely possible. And so one of the grants, the SBIR grant, will also do a toxicity study, so we can really narrow in on the dose range. But I agree. I mean, when everyone sees this, it's like, that's too good to be true. How can this plant really do all these things? And it's, you know, it's sold as a supplement on the internet, and you see all these things, and it's all these testimonials about how moringa changed my life and saved my life. And so I kind of had the same feeling, but then when I started looking at the science and the chemistry, I was much more convinced.
And then I was thinking, do you know how can moringa as a plant get all these great nutrients compared to, say, like the baobab trees, you know, when it's in poor soil? So, like, how does that work? That it gets many more
Right,
nutrients and things.
and the most baffling part is the protein, is that it's not a nitrogen-fixing plant. It's not a legume. So how is it getting nitrogen? So we did try and look into that, and it was a project that I really wish I could have spent more time on. But one hypothesis is that it has endophytic bacteria that can actually fix nitrogen from the air. And because so even in poor soils, there's no other really way to explain it. So as a non-legume plant, by dry weight, it's 27% protein, which is the highest of any known plant. So something is going on, like you said, if it's growing in sandy soils. But it also has a high capacity to uptake nutrients, which can also be a detriment if you're growing in soils with metals and contaminants as well. It will also take those up. So that is a danger, and that could be, so you really have to, you know, test for safety. And they've done some work with using it for that reason, for bioremediation, where you actually suck up lead and stuff from the soil and then burn it off to clean up the soil. So we'll definitely have to look at the land and where it's grown and do some of these studies that's where it's being produced in Kenya.
Do you intend to do any more research with using moringa as part of a complementary food for infants?
I don't have any direct plans, just because it's not in the, you know, I'm so excited about it with all these opportunities. But if there's an easy way I can get in on, you know, a trial that's already being done or something like that, or adding it into a food, then that would be possible. And there's already a lot of preliminary studies and data that's shown that it's really beneficial. And since it's a food, it's much easier, you know. So not right now, but hopefully I'll be in Kenya for a month, and I'll be back, and I'm going to just kind of get the lay of the land and see how things are going. So.
You mentioned it can be used to promote milk production in animals.
Mm-hmm.
Is it similar for humans?
There were a couple of studies that showed increased lactation in humans, but I would like to see more studies done in the States. There's been no clinical studies on moringa in the United States. So not that I'm just more skeptical of some of the publications and the journals that I found, but there have been 2 or 3 that have shown that it increases lactation in humans as well. I don't know how we are on time. If I went too fast or too slow, maybe it was too fast.
So I was going to ask Malloy about availability in Bangladesh, because drumstick is moringa. I don't know. Drumstick, the vegetable, is moringa, and I think it's the stalk part.
Yeah.
So, do you know to what extent that's consumed by lower income households and by young children, or not? Anything about it?
I I don't know actually.
Yeah.
But in Bangladesh, there is another plant which is known as neem.
Yeah.
Yeah, and which is consumed by a lot of people for its medicinal purposes. But I don't know much about it.
Is it related?
It's not moringa.
No.
No, it's different. It's a different family. And it's used a lot as an insecticide, actually, the neem oil. But it's from people who I've met that have come from India, a lot of people said they just have one tree in the backyard and they'll go out and add it into their foods as they cook. And so.
Yeah, and also, for example, if you suffer from chicken pox, so they make paste out of those leaves and just apply those paste to blisters. And I know many children who actually extract, boil the leaves and extract something from the leaves, and the children are fed that extract almost every day.
And
sometimes.
is that moringa or is that the other one?
No, this is the other one.
The other one.
And it's pretty common in Ghana too. There's been a lot of noise about it, about the health benefits. So a lot of people consume different ways.
The leaves mainly, or the seeds?
The leaves,
yeah.
Are most of the beneficial compounds damaged through heat, through cooking? Is it best to consume it dried or?
So nutrient-wise, some of you might have a better idea of that, or even Beth Mitchum at the Post-Harvest Center, where they look at that, and certainly at a certain temperature. The one that's most vulnerable that I know of is vitamin C. So that definitely decreases in the dried versus the fresh. But everything else, because you're concentrating it so much and losing the water, you're increasing usually 3 times the amount of nutrients that are there. So the isothiocyanates are more stable than they are in broccoli, and so they can withstand some temperature. So we'll have to also monitor that as we process it. And then some polyphenols are more prone to heat degradation. So everything is to some degree, but there's nothing that's rapidly going to degrade, Mm-hmm.
So talking about the active compound.
Yes.
What about the supplement, the powders that are sold in the market because you buy them already grounded?
Yeah.
And what about because you say that there is a degradation over time.
Right.
So someone who buys it for the daily use, how is it about this?
So I didn't do any experimental comparisons of my moringa concentrate versus the powders that are sold. But what I would think is that the powders that are sold are going to have similar amounts of nutrients as the moringa concentrate that we're processing. It's just that the moringa concentrate we're making would have higher levels of isothiocyanates. So I think there's still a great deal of health benefit in consuming what's already on the market. But ideally what we'd like is to alter the processing so even those companies that are producing it would produce it in a different way that would harness these specific compounds that need that initial crushing to convert. But I still think, the polyphenols themselves are really beneficial, and then this high nutrient content and the amino acids is also good. So we're trying to kind of have a better product in some ways, and that hopefully in the U.S. we can patent and really make money off of it, and then in the developing world just go out to these villages and say, hey, put it in your mortar and pestle, and then lay it out on a screen and dry it, and then put it into a powder. So it's kind of as simple as that.
The second question is about going like Africa or Asia, because the leaves are consumed.
Mm-hmm.
What could we expect from someone who consumes the leaves? Is it because generally they will boil it and cook it as a sauce or something like this? So is it any expectation about?
Yeah, you would definitely want to recommend that it's. So what I've seen in Africa was it was kind of being promoted as a powder form that you would, after you were done cooking, you would sprinkle it on top. So you wouldn't boil it a lot. But I don't know in India and Southeast Asia, where they actually cook with it more, I assume that they do boil it. So if you've ever had masala dosa, which is a South Indian dish, there's a sambar soup, and they have the pods in it, and I'm pretty sure they boil those the whole time because they're even still kind of hard and fibrous on the outside and then have a little bit of flavor inside. So with that, you probably are losing more of the nutrients.
I have a question about the broccoli, actually, the sulfur
Okay.
things. If you know the recommended way to prepare it to get the most out of it?
Yeah, and there's, if you give me your email, I can send. There's a couple really good papers that have looked at microwaving and steaming and all blanching and boiling and all the different ways and how fine to chop it and all of these things. I mean, they've really done a lot of work on it. But I think fresh is probably the best, or lightly steamed, where you don't, I think with any vegetable, it doesn't leach out into the water. What I do with mine, I like it, but I'll just put a little bit in a frying pan, and then I drink the water afterwards that's in the pan. Especially after doing all this research. So.
And those papers are looking at the sulforaphane?
Yeah, sulforaphane. Yeah, there's a lot more. And so what's really been nice about this project is we can just piggyback on broccoli. And there's been clinical trials and all these studies. So I can basically read an assay that was done with sulforaphane. So, for instance, sulforaphane is known to trigger these detoxifying enzymes, and that's why it's so good for cancer prevention. And I looked at the study, and I'm like, well, it has the same pharmacophore, so the same active part of the molecule, and I pretty much can guarantee if I test the Moringa isothiocyanates, it'll have the same activity, and that's what we found. So Moringa has direct antioxidant activity where it captures free radicals mainly from the polyphenols, and then indirect antioxidant activity from the isothiocyanates. So it's nice when you have that prelude to go off of,
I heard the production is somewhat challenging, or the processing. Do you know about that, or is it just a matter of knowing how to do?
I've only seen it on a really small scale, just in the villages where they get it, lay it out in the sun usually, and then grind it. I made it myself when I was there, so it's pretty simple. The leaves, they kind of have those fronds, and you can just pull it really easily off with your hands, and they stay together really nicely. They're really firm. They don't wilt as much or anything like that. And I know in Israel they have a plow that goes through the fields and just grinds it all up, like very large-scale industrial processing of it.
So can you grow it in the United States?
Yeah, they have farms in Florida, in Kenya, in Southern California, and a lot in Hawaii. But the main guy I've worked with from Los Angeles, they do grow them in the greenhouse over the winter, and he doesn't grow as much of it in the winter, so, or whatever winter is in LA. But it's it's definitely colder. So yeah, it can be done, and that's one hope as well. If I can transfer to UC Davis, that I would do some work here in the greenhouses as well. Because there's always so much you can play with in terms of the variety of the plant. When do you harvest it? We did some studies that looked at the sprouts versus the full leaves. Same with broccoli sprouts. The sprouts have higher concentration of these compounds. So all of those techniques could be looked at. But I think I want to just move forward and get to the animal studies, and then see if we can hopefully set up some work with either infants or humans in Kenya or in the States.
So there are many species of Moringa.
Yeah, there's about 13 species, and Moringa oleifera is the one that's most, it's one of two that's cultivated. The others are all wild. And there's some that have only been seen once or twice, and some of them look very different. They're known as ghost trees, and they grow like in Madagascar in the complete desert, where you get this white trunk with basically no leaves at all. And that would be yet another thing I'd like to look at, is I don't think anyone's done any phytochemical analysis of these other species.
Okay, to add to those. So in Bangladesh, like nobody, I mean, I don't know that anybody eats the leaves of moringa, but the pods, they can be used as vegetables.
Do you have any idea why they don't eat the leaves?
It's not like in the
I was
culture.
saying about Chajna.
Yeah.
Yes, leaves are also taken by some of the people. Yeah.
Okay. Is it a regional thing?
What do you?
In different parts of the country where it's more likely?
No, it's basically in the rural area, but it's not that popularly cultivated, but it's available in some rural areas, and rural people usually take the leaves and they either fry it or sometimes they make some mash out of it and then eat with rice. And the long stalks, they are usually cooked with other kind of like fish or beans, these kind of things. But it's very popular vegetables in rural area and also in city area.
Are the trees wild or cultivated?
Some are wild, some are cultivated. But it's not cultivated in a large scale.
Like in the northern part of Bangladesh, where we implement the research, so you can find, during the seasons, you can find that pods of moringa in the market. So, I never heard of anyone eating leaves, so
But the potential is there.
the potential is there because like eating leaves is in the culture, so, like neem.
Or other leaves, yeah.
And the taste is good. It's not like neem is bitter, but it's not. The taste is good. It's not sweet, but little sweetish.
Yeah, that's an important thing to mention.
Well,
It's a safe for eating.
The property that you mentioned about taking up toxic metals from the soils could be very important and interesting to explore in Bangladesh because the soils there are high in many areas in arsenic, cadmium, and I just learned from Steve Luby lead.
Wow.
And in fact, in the Wash Benefits trial, they analyzed the rice that people were eating, and the lead is in the rice, and it's actually internal to the grain. So it's been taken up by the rice plant and internalized.
Yeah, the same is for arsenic. See, some of the rice varieties, arsenic is there. So, I mean, there are some research in Bangladesh which used moringa plants to take up arsenic.
Did it work?
I don't know the results.
Well, I'm just wondering if they grow in the kind of the paddy areas where the rice would be grown.
I
Can
don't
you
know.
grow
I
it
think
in-
it's too
sensitive to flooding?
Yeah, it really likes dry, sandy soil. So in the paddies, it would be hard for rice, but maybe other areas would
Or vegetables or
Usually grown at the yard of the houses, just for sale.
Yeah, which isn't where the rice is grown.
but I know a lot of other brassica species, like just mustard and stuff like that, has also been used for phytoremediation, and maybe some of that is more amenable to wet soils.
Yeah, mustard is grown on many places between the two areas.
I'm sorry I was late.
No
I had
problem.
another workshop I was speaking at, so I couldn't get that much. But I heard Carrie talk about this in August, so I got the first part, and I'm very happy that she was able to do the presentation for our seminar today. And I hope you'll keep us updated on what you learned. Come back and give us some more data.
Okay.
But please join me in thanking Carrie for her very useful seminar.